Hello students, welcome to CBSE NCERT Solutions by Vedantu. I am Ms. Gopika, your own biology math teacher. So yesterday we have done excretory product and the elimination part 1, where we understood about the different types of mechanisms, and also we learned about what is the basic, okay, what are the excretory organs in different animals, and we also learned about the structure, human excretory system. We understood how their modifications are, what are present. All this we have understood in yesterday's class. Today we are going to go into the main portion, that is urine formation. Okay, so we will learn urine formation. We'll also read NCERT of what we did yesterday, that is your RAS mechanism, ADH mechanism, and ANF mechanism. Okay, before that, let me just check the voice and the video. One second. For that, let me just check the voice and the video. Okay, it's perfectly audible. Okay, yes. So happy Navratri to all the students who are celebrating it. Okay, wishing all of you a very, very prosperous and happy Navratri this year. And let your, whatever dreams it is, be it competitive exam, be it your board exams, everything, let it be successful. And yes, so we will go into the second part of our excretory product and elimination, students, that is our urine formation. Okay, so urine formation actually takes place in 3 steps. Okay, so we are going to learn each of the steps. So let's go one by one. Urine formation involves 3 main processes like glomerular filtration, reabsorption, and secretion that takes place in different parts of the nephron. First step, okay, one second, yeah. The first step in urine formation is the filtration of the blood. Obviously, that should be the first job, because in the blood, the toxic substances are going to be there, like your urea. So that has to be first filtered out, forming your GF. Yesterday we have spoke, glomerular filtration, which is carried out by the glomerulus, and it is called as glomerular filtration. On an average, 1100 to 1200 ml of the blood is filtered by the kidney per minute. So you can imagine how much ml is going around. 1100 to 1200 ml, which constitute roughly to 1/5 of the blood pumped out by each ventricles of the heart in a minute. Yes or no? So almost this much is going out. So students, how much it is pumped into the cardiac? Just tell me how much blood do the heart pump. Okay, if they're telling that this is 1/5, so you just have to multiply into 5. No, so approximately around 5.5 ml. Right, 5.5 ml is going to be pumped, out of which 1100 to 1200 ml is going to be filtered per minute from the kidney, by the kidney. Okay, now the glomerular blood capillary, yes, very good. The glomerular capillary blood pressure causes the filtration of blood through 3 layers. So students, like this we have studied, like diffusion membrane, like breathing we have studied about the respiratory membrane. So because of the pressure, okay, same way, because of the pressure gradient in breathing, the gas is going through 3 layers. Okay, here also because of the pressure, etc., the blood is going through 3 layers. What are the 3 layers? The endothelium of the glomerular blood vessel, the epithelium of Bowman's capsule, and the basement membrane between the 2 layers. Okay, so again here also there is non-cellular 1 layer and 2 cellular layers. Endothelium of the glomerulus. Okay, so this is the glomerulus. Okay, I think I'll draw it down. Okay, the epithelial cells of the Bowman's capsule, called podocytes, are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pores. So students, imagine something like this. Okay, all of you hold hands like this, no? See, when you hold your hands like this, can you see some spaces in between? I think all of you can see the spaces in between my hands. Now, these kind of pattern is how your podocytes are going to be arranged. So they are going to be arranged in an intricate manner, something like this. Okay, so that there will be spaces in between, which are called as a slit pore or the filtration pore. Okay, filtration pore, slits. Blood is filtered so finely through this membrane that almost all the constituents of plasma, except the protein, passes into the lumen of the blood vessel. So what are they telling? Blood is filtered so finely through this membrane that almost all the constituent of plasma, except proteins, because proteins are larger substance, so they will not pass. Okay, they will not pass. Therefore, it is considered as ultrafiltration. Okay, therefore it is considered as ultrafiltration. So far clear? Yes or no? Tell me. Okay. So students, how does this entire urine formation happens? Let's say this is the glomerulus. Okay, this is your glomerulus, like this. Okay, this is your tubes. If you want, we can do the cross sectioning like this, like make it condensed. Okay, like this. Okay, assume this is our glomerulus. Okay, I mean this is the Bowman's capsule or Malpighian corpus. So this is your glomerulus. Okay, now this is the blood vessel which is going downwards. Got it? Yes. Done. So now students, exactly what is going to happen? The first step is going to be glomerular filtration. That means the blood that is coming from your efferent arterioles is going to pass through the glomerulus and cross through the Bowman's capsule. So here, technically speaking, here is going to be your first step of your urine formation, which is called as glomerular filtration. Got it? Yes or no? All of you in the chat, tell me, did you understand this? Glomerular filtration happens when the blood is passing through the glomerulus to the Bowman's capsule. Okay. Second thing, okay, all of you mark your attendance as well. Like should keep increasing. Second step is going to be tubular secretion and tubular reabsorption we have. Okay. Second is going to be, students, tubular reabsorption, where your tubules are going to give out whatever they are not necessary. Okay. Whatever is not necessary for them, they are going to give, which is called as tubular reabsorption. Okay, tubular reabsorption. Got it? That is the second part. That means whatever the tubules require or whatever is required for the, like, let's say expelling out in the urine, that will be taken. Rest everything we will give it to the blood. Now the third step is going to be, I think I should have written it here. 1 second, students. Okay, I'll just write it this side. Okay. Yes, the second step is going to be tubular reabsorption. Okay, reabsorption. Yes. Now the third step is going to be, now blood would have got so many necessary, unnecessary things. Now blood will be like, I don't want this. This should be going back into as in the form of urine. So it will give back, which is called as tubular secretions. Okay, tubular secretions. Got it? These are the three main steps. Got it? These are the three main steps of your entire urine formation. Okay, your entire urine formation. Now, so this is the urine formation, 3 steps. Now students, the first step is going to be glomerular filtration or ultrafiltration. In this, what is going to happen? Let's say this is your Bowman's capsule. Okay, this is your Bowman's capsule. These podocytes are going to be arranged in an intricate manner, as mentioned. No, this is the intricate manner. Okay, they are going to be arranged in an intricate manner like this, and they will continue. And this is going to happen. So this is how it is going to happen. Then let's say, yes, urine formation, very good. Okay, let's say, I don't think everything will fit in here, students, but let's try. Okay, trying is always important. Yes or no? So let's say that's your glomerulus. I've separated it and drawn, otherwise you will not understand the 3 layers. Okay, you will not understand the 3 layers. So let's say if I separated this is the glomerulus. So what are the 3 layers mentioned? Can any of you tell me what are the 3 layers mentioned? First was endothelium. Okay, endothelium of your glomerulus. Next one was epithelium. Epithelia of Bowman's capsule, which is called as podocyte. And third one was basement membrane. Yes or no? Basement membrane. It is on Sunday, Tanuja, because I didn't get slot, so it's on Sunday. Okay, it will be on Sunday. So see you all on Sunday 5 p.m. Students, my neck has, I don't know, something has happened. I have to keep holding my hand sometimes, so it pains. Okay, so endothelium of the glomerulus. So this is your Bowman's capsule. This is the glomerulus. So this is going to be the endothelium. Okay, endothelium of the glomerulus, a layer like this. Okay, cells, no? So like this it will be. Endothelium of the glomerulus. Then epithelium. These are the podocytes. Epithelium of the Bowman's capsule means this only, podocytes. Very important, podocytes. Now between these podocytes, what you're seeing, no, the spaces, these are going to be your slit, filtration slits. Okay, filtration slits. Yes. Got it? Yes or no? Clear? Podocytes and filtration slits are actually spaces. I think I want to show you guys a diagram, but I think I will not be able to put the PDF. Let's just try, okay? Let's just try. Right. Got it. Now let me try to find an image. Students, look here. Is this the best image? I think this is too complex. Okay, I think this is the most simplest image I can find. Okay, all of you look here. This is my phone. Okay, phone screen. You can see, no, the ones that you're seeing, the darkish yellow ones, those are your, okay, this one, okay, these are your podocytes. These are your podocytes. Got it? Between this, what you're seeing is your filtration membrane, like this slits, no, this is your filtration slits. Then as you go down, you can see your glomerula. Here you see glomerula. Okay, and as you move this side, see here, you can see the endothelium. Okay, you can see the endothelium. So this is your 3 layers. My God, it is your 3 layers. So in between these 3 layers, there is going to be a basement membrane. Down you will see endothelium, and top you will see podocytes, and the between space is called as a filtration slit. Okay, if you want, I can draw this diagram only. If it is more easier to understand, I think I'll draw this diagram. Oh, I zoomed in so much that the diagram only is not working. Okay, chalo, I'll draw this diagram here. 1 second, students. I need to find space to draw this diagram. Okay, wait, I'm going to draw the same diagram here, so you will also be able to understand. Okay. Okay, these are your podocytes. Okay, they're not exactly like this. This is just a very diagrammatic representation to make you understand. This is how it will be, intricate manner ones. The one I drew, it's kind of similar. Okay, now here they have drawn the glomerular filtration, and here they have drew the endothelium. Okay. Yes, that. Okay, now let's label it so you'll understand. This is your podocytes. Okay, this is your filtration slits. Okay, this is the basement membrane. Okay, basement membrane. This is the endothelium. It's actually endothelium of the capillaries, but we write endothelium of glomerulus also, no problem. Okay, because again, they're all blood. They're tuft of capillaries only, you know. So is this diagram better? This is the most simplest diagram. Okay, simple baby diagram this is. All of you tell me in the chats. Fine. Yeah, now is it easy? Now is it easy? It might look like a tribal's, head, like cap or something, but it is not. Okay. But yeah, this is the simplest diagram for you to understand this. So these 3 layers are going to help for the filtration. Okay, help for the glomerular filtration. So around students 180 liter of glomerular filtrate is going to be filtered out of which only 1 to 1.5 liters of urine only will be expelled out in a day. Understood? 1 to 1.5 liters. Yes, now see here what I told. The amount of filtrate formed by the kidney per minute is called as the GFR, which is called as the glomerular filtration rate. GFR in a healthy way is an in an healthy individual is approximately 125 ml per minute. That is 180 liters per day. But out of this 180 liters, there is only going to be 1 to 1.5 liters of urine that will be expelled out. Now the kidney have built-in mechanism for regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus. Yesterday I taught you juxtaglomerular apparatus. No, tell me what they are going to release. JGA is a special sensitive region formed by the cellular modification in the distal convoluted tubule and the afferent arteriole. So in the distal convoluted tubule, what is going to be formed? Macula densa is going to be formed. Okay, in the DCT, macula densa is going to be formed. Again, it's a sensor cell who will identify that GFR as an issue, and afferent arterioles, what is going to happen, students? They are going to form JG cells. Okay, afferent arterioles will modify to form JG cells. Very good. And this JG cells will release renin. This JG cells will release renin. Okay. Now, JG cells is a special sensitive region formed by the cellular modifications in the distal convoluted tubule. The afferent arterioles at the location of their contact, a fall in GFR can activate the JG cells to release renin, which will stimulate the glomerular blood flow, and thereby GFR go back to normal. Okay, but the thing is, I told you, renin cannot do the job alone. So renin will take the help of, whose help will it take? It will take the help of angiotensinogen and convert it to angiotensin 1, angiotensin 2, and then do the rest of the function. Now, a comparison of the volume, okay, a comparison of the volume of filtrate formed per day, that is 180 liters per day, with that of the urine released, suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. Yes or no, students? All of you agree or not? Okay, that 99% is going to be reabsorbed because 180 liters, okay, because 180 liters is formed per day. Out of that, only 1 liter is going out. Okay, 1 liter is going out. That means 99%, this is the reabsorption is going to happen. Correct? Okay. I did, but it's not reducing. I think it's my nerve. Okay, one of the nerves have got caught, or whether I slept, I did not sleep properly or something like that. Neck pain is the worst, students, because neck pain leads to head pain leads to eye pain leads to ear pain. So it's the entire facial features will start paining. So this process is called as reabsorption. Okay, this process is called as reabsorption. The tubular epithelial cells in different segments of the nephron perform this neither by active or passive mechanism. Okay. So the tubular epithelial cells in different segments of the nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acids, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by passive transport. Okay, so here, students, for example, substances like glucose, amino acid and sodium in the filtrate will be reabsorbed actively. This question has come previously. What is reabsorbed actively? What is reabsorbed passively? But nitrogenous waste will be absorbed passive. Okay, passive. Now, reabsorption of water also occurs passively in the initial segments of the nephron. During urine formation, the tubular cells secrete substances like H+, K+, and ammonia into the filtrate. During urine formation, the tubular cells secrete these things, H+, potassium, ammonia into the filtrate. Okay, into the filtrate. Now, tubular secretion is also an important step in urine formation, helps to maintain the ionic acid content. Now, very important functions of tubules. Okay, proximal convoluted tubules. PCT is lined by simple cuboidal brush border epithelium. What is it lined by? PCT. Okay, PCT is lined by okay, this is your Bowman's capsule. Okay, let's just draw PCT directly. Drawing Bowman's capsule, I've just learned the diagram so well. Okay, This is your PCT. Okay, now here they have brush border. Okay, wait, brush border. Since it's too difficult to draw here, did I leave space here? Yeah, I left space here. Okay, do this. Yes, do this. Okay, now they're telling they have brush border cuboidal epithelium. So this is brush border. Okay, this is brush border cuboidal epithelium. What is brush border going to do, students? It is going to increase the surface area. Okay, so more reabsorption happens. Got it? More reabsorption happens. Very good. Most of you are telling the answer. So this is going to be brush border cuboidal epithelium. Where is it present? This epithelium is present in the PCT. Okay, epithelium is present in the PCT. Now students, PCT does maximum reabsorption. Up to 70 to 80% reabsorption is done by PCT. Done? Yes, very good. More reabsorption. So here what is going to happen? Look here, around 70 to 80%, which increases the surface area. As I told you, nearly all essential nutrients and 70 to 80% of the electrolytes and water are reabsorbed by this segment. Okay, understood? They are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ions, ammonia, potassium ions into the filtrate, and by absorption of HCO3 minus from it. So what they will do, they will give all this to the filtrate, and then whenever required they will take HCO3 minus. Okay, HCO3 minus. So here what is going to happen, this proximal convoluted tubule PCT also helps to maintain the pH and ionic balance of the body fluids by selective secretion. Got it, all of you? Yes or no? So PCT, okay, does 70 to 80%. They do selective secretion and selective absorption, reabsorption. Okay, selective reabsorption. Yes, done? Clear or not? Done. Okay, perfect. Exactly, please go. I'm not able to teach actually. My head is paining and my neck is also paining. Okay, I thought it was fine, but it's not now. Now when I'm sitting, I'm not able to be. Okay, so PCT is going to do selective secretion and selective reabsorption. Okay, so here 70 to 80% of the electrolytes and water are reabsorbed by this segment. PCT also helps to maintain pH and ionic balance. Okay, this is also clear. Secretion is going to happen. Secretion of what? Hydrogen, ammonia. Okay, hydrogen, ammonia, and potassium ions. Potassium ions. Now into the filtrate. Okay, it is going into the filtrate. And from the filtrate, what are you taking? From the filtrate, you are going to take HCO3 minus. HCO3 minus. That next tube, Henle's tube. Okay, next tube is going to be Henle's tube. Henle's loop is very important for the maintenance of the concentration of the urine. So reabsorption is minimum in its ascending limb. However, this region plays a significant role in maintenance of high osmolarity of medullary interstitial fluid. So students, main job of loop of Henle is to participate in countercurrent mechanism. Now all of you, I'll teach you countercurrent mechanism. Now countercurrent mechanism is going to help with the osmolarity. Osmolarity is going to go from 300 up to 1200 ml osmolarity is going to happen. Okay, that is going to happen with the help of Henle's loop. So if loop of Henle is not there, then the countercurrent mechanism also will not happen. So the descending limb of loop of Henle is permeable to water but almost impermeable to electrolyte. Who? Descending limb. So descending limb, so D is permeable to water. Imagine your friend, okay, Dikshi is permeable to water. That means she loves drinking water, but she does not like eating any of the electrolytes. She does not drink anything, okay, any of the electrolytes. So here D is going to be permeable to water. Yes or no? D is going to be permeable to water. That means descending limb is going to be permeable to water, and they are going to be impermeable to electrolyte. So one is permeable to water means that person itself will not be permeable to electrolyte. So they are going to be ulta. Who tell? Ascending limb and descending limb. Understood? Yes or no? Chalo. Now this concentrates the filtrate as it moves down. So students, this is the descending limb. Okay, this is the descending limb. This is the ascending limb. So as it is going down, what is going to happen? It is going to be permeable to water. No. So this concentrates the filtrate as it moves down. It is going to keep concentrating because continuously electrolytes have been inside. Okay, electrolytes are here. So what do you expect in the ascending limb? Ascending limb, students. In the ascending limb, what you're going to expect is, in the ascending limb, it is going to be permeable to electrolyte and impermeable to water. Okay, so here you can see this concentrates. Okay, the ascending limb is impermeable to water but allows transport of electrolyte actively or passively. Okay, actively or passively. Therefore, as the concentrated filtrate pass upward, it gets diluted due to the passage of electrolyte to the medullary. So here, what is going to happen? These electrolytes, let's use another color. These electrolytes are going to be taken into the interstitial or medullary fluid. Okay, to your medullary fluid. Exactly, magic. Okay, medullary fluid. Done. Now when you learn counter current mechanism, you'll understand. Okay. Now distal convoluted tubule, student, DCT. This is conditional reabsorption. Okay, conditional reabsorption of sodium and water takes place in this segment. So conditional reabsorption means what? Only when it is required. Okay, only when it is required. Only when it is required, it is going to happen. Got it? When, like, if it is not necessary that every time reabsorption happens. When they think that it is really necessary to do reabsorption, they have to do reabsorption. Understood? Yes or no? 1 second, students. Okay. Because this neck is paining, upon that hair also will cause the pain. Okay, yeah. Chalo. Homeostasis is the maintenance of your body temperature. Homeostasis is maintenance of your body temperature. Okay. Yes. Now students, DCT is conditional reabsorption of sodium and water takes place in the segment. DCT is also capable. Okay. DCT is also capable of reabsorption of HCO3 and selective secretion of hydrogen, potassium and ammonia to maintain the pH and the sodium potassium balance. Understood? Clear or not? Yes? 1 second. Done. Okay. Now conditional reabsorption of sodium and water takes place. Okay, conditional reabsorption of sodium and water takes place. So the students, with the help of DCT only, we are going to help or make most of the mechanism work as well. Okay, why? Because we will release something. Okay, release something and we will help this person telling that, okay, now let's imagine, like let's take an example of Mohit. Okay, Mohit has high blood pressure. Okay, Mohit has high blood pressure. So if Mohit's high blood pressure is high, we expect that ANF mechanism works. Yes or no? Atrial natriuretic factor works because the heart will be able to feel the pressure of how the blood is flowing. So if the pressure of the blood is too high through the arteries, then the ANF mechanism is going to happen and it is going to lower the BP. Now at the same time, if RAS mechanism have to happen, Okay, RAS mechanism have to happen. Okay, so if RAS mechanism happens, students, where the BP is, BP is low, or let's say blood volume is low, then we are going to tell that, okay, do you know what, we will take the help of distal convoluted tubule and our collecting duct to actually reabsorb sodium and water. Okay, got it? Yes. Now Mohit, you can check or download it from Vedantu website. Okay, I do not have a thing in my hand. Okay, now students, look here, this is going to be your PCT. Okay, this is your PCT. So PCT is going to do conditional reabsorption and secretion. Okay, so you can see H plus and NH3 is going into the filtrate, and these things are going outside. Okay, going outside the filtrate. And then when you come down here, this is going to be permeable to water, and this is going to be permeable to electrolyte. Actually, HCO3, NaCl, this H plus, this here, this arrow mark is here. Okay, this should have been here, K plus. Okay, K plus should have been here. Got it? Example of animals. Yeah, perfect. Okay, very smart. Okay, so here, students, this is going to be permeable to water. So as water is going to go down, the electrolyte is going to get very much concentrated here, and as it is going up, what is going to happen? The electrolyte will be removed by the ascending limb, and it will be added into the medullary interstitium. So this is your medullary fluid here. Now this you will study in countercurrent mechanism. Now from the collecting duct, some amount of urea, okay, from collecting duct, some amount of urea is going to be taken here, back here again, coming to learn in medullary, that is your countercurrent mechanism. Now students, here so you can see NaCl, H2O, HCO3, K plus, H plus, all this is going to have secretions and reabsorptions. Okay, students, I cannot block, so you people please block. Okay, yes. So here it is going to the collecting duct. Okay, now collecting duct. This long duct extends from the cortex of the kidney to the inner parts of the medulla. Okay, to the inner parts of the medulla. Large amount of water could be, okay, large amount of water could be reabsorbed from this region to produce a concentrated urine. Okay, a concentrated urine. Got it? Now this segment allows passage of small amounts of urine, okay, small amounts of urea into the medullary interstitium to keep up with the osmolarity. It also plays a role in maintenance of pH and ionic balance of the blood by selective secretion of H plus and potassium ions. Okay, H plus and potassium ions. So collecting duct, this is the long duct which extends from the cortex of the kidney to the inner part of the medulla. Large amounts, okay, large amounts of water could be reabsorbed from this region to produce concentrated urine. So when the urine is going, so what we will do to take maximum water, because we do not want to waste any more water. So what we will do, we will tell that, okay, don't worry, collecting duct is going to reabsorb all the water. So this segment, okay, this segment allows passage of small amount of urea into the medullary interstitium to keep up the osmolarity. Got it? Now, mechanism of concentration of filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta plays a significant role in this. The flow of filtrate in two limbs of the Henle's loop is in the opposite direction. So see students, the flow of filtrate in two limbs, okay, of the Henle's loop is going to be in, opposite direction, and thus forms the counter current. The flow of blood through the two limbs of the vasa recta also in the counter current pattern. The proximity between Henle's loop, okay, The proximity between Henle's loop and vasa recta as well as the countercurrent in them help in maintaining and increasing the osmolarity towards the inner medullary interstitium, that is from 300 osmolarity to 1200. So students, here this countercurrent mechanism, you can all look at the image here. Okay, now you can see here that, see, in this it's a little complicated. Why I'll tell you it's a little complicated, because here this is the vasa recta, this is the nephron that is showing. Okay, so osmolarity is the basic balance, okay, salt water balance in your urine. If salt water balance is not there in your urine, then what happens, maybe there will be lot of water in the urine, or there will be too much of electrolytes in the urine. So to balance this, there should be an osmolarity. That means to maintain the salt and water balance, that is called as your osmolarity. For that we have countercurrent mechanism. Okay, countercurrent mechanism. Now here students, here if you see here in this diagram, this is your vasa recta. Okay, so here you can see as it is coming down, NaCl is taken up and it is moving downwards all the way. But this is a very confusing diagram, so instead of that we will draw our own diagram. Just look here, this is your loop of Henle. Okay, Mohit, why are you going to visit her? By knowing her location. This is your loop of Henle. Okay, this is vasa recta. Vasa recta. Okay, now this loop of Henle, obviously here DCT is going to come, collecting duct is going to come. Okay, I'm just drawing a very average diagram, okay, below average diagram, so that I can, because if I draw that you will not understand. Okay, you will not understand, so that's why I'm drawing this diagram. So this is your collecting duct. Collecting duct. Okay, fine. Okay, now students, look here, what is going to happen? This is your descending loop of Henle. Okay, this is your ascending loop of Henle. Okay, this is going to be ascending vasa recta, this is going to be descending vasa recta. Correct? Now between these people, okay, between these people, students, this portion is called as the medullary interstitium. Okay, this is going to be called as the medullary interstitium, this portion. Now what is going to happen? DOH, okay, that is your descending loop of Henle, is permeable to water but impermeable to electrolyte. Okay, so water will keep coming out, doesn't matter, it will go all the way till here. Here NaCl, they told, no, just before this paragraph they had told NaCl will be secreted into the medullary interstitium. That will be picked up, okay, that will be picked up by your descending loop vasa recta. Okay, that will be okay, that will be, sorry ma'am, since okay, fine. Yes, so students, this NaCl will be picked up by the descending vasa recta, carried all the way. Okay, it will go all the way up to the ascending vasa recta and it will deposit it into the medullary interstitium. So now here continuously this is going to happen and the concentration of NaCl is going to increase and this solution is going to become a hypertonic solution. Hypertonic solution. And with NaCl, I told you that some amount of urea from the collecting duct is also going to come into the medullary interstitium. Some amount of urea as well. So NaCl plus urea, okay, NaCl plus urea will increase the concentration here to make it a hypertonic solution, because of which, students, more water from the fluid is going to come out. Yes or no? If the solution is hypertonic, definitely water is going to ooze out from a region of higher concentration to a lower concentration. Correct? Is this clear? Amazing diagram. Okay, done. So So students, this is usually done during the stress mechanism. That means if a person has not consumed enough water, if a person has, like how you people do during Navratri, we all do fasting. If all that is happening, then there is high chance that the person will not have enough water in the body. So during this situation, your juxtamedullary nephron is going to do this job. Okay, juxtamedullary nephron, because juxtamedullary nephron has vasa recta, if you all remember, and they are going to have long loop of Henle. This juxtamedullary nephron, students, is going to do this countercurrent mechanism with a loop of Henle and vasa recta, where they will try to reabsorb as much as water from the tubule so that the urine that is coming out will be concentrated. Okay, yes, we do, sometimes we do fasting. Okay, like not every time. Now that since I'm teaching, I cannot do it because it drains a lot of energy, but yeah, if it is necessary, we do it. Okay, so students, here what happens, the urine formed will be concentrated. The urine form will be concentrated. Understood? Yes or no? So if loop of Henle is not there, then the urine will become diluted. Clear or not, students? Yes, all of you? Students, my neck pain has gotten a little severe. I'll try to apply something and come back to the class. Okay, just give me 10-15 minutes. I'll come back. I'm not able to teach. Okay, max 20-25. I'll come back. Okay, because my neck pain has got very severe and I'm not able to teach. I told you guys up to 7:25 max, and you people are crying in the chats. I told you guys up to 7:25. So fight, everything is over. But small kids. Okay, I hope countercurrent mechanism was clear. Okay. Yes, countercurrent mechanism was clear. Now let's move to the next one. So students, in the medullary interstitium, presence of such interstitial gradient help in an easy passage of water from collecting tube, thereby concentrating the urine. So as I told you, this usually happens in stress situation. Okay, this usually happens in stress situation, and that was helping to make sure that the urine gets concentrated. Okay, urine gets concentrated. Chalo. Now, moving on. Regulation of kidney function. So we have already learned this yesterday, so it's going to be like a revision for all of you. Okay, students. Yeah. People are biggest kids I've ever seen. Students, I forgot my glasses. One second. Yeah. People are not liking the video. Okay, why is that so? All of you are very excited to tell hello. Why can't you like the video? The functioning of kidney is efficiently monitored and regulated by hormonal feedback mechanism involving the hypothalamus, JGA, and certain extent the heart. So here they're talking about hypothalamus because we need to release vasopressin. Yes or no? We need to release vasopressin, which is an anti-diuretic hormone, which is going to help in whenever there is a lack of water, whenever there's lots of water, this is going to happen. Okay. Chalo. Now, an excessive loss of fluid from the body can activate these receptors which stimulate hypothalamus. For those students who did not listen yesterday's class, there is two mechanisms. Okay, I'm not going to do the whole mechanism, but I'll just tell you a gist of it. Okay, gist of it. It is ADH mechanism. Okay, that means whenever there is a lot of loss of water. Okay, loss of water, it could be because of diabetes mellitus or anything like that. Lot of water is lost. Then what is going to happen? Then our body will activate hypothalamus, and this hypothalamus will release or activate the posterior pituitary to release vasopressin. Okay, vasopressin or ADH hormone. This ADH hormone is going to do two things. First thing, exogenously inform you, telling that, hey, Mohit, drink some water, man. You haven't drank water from the morning. So, exogenously means it is going to activate your thirst mechanism. Telling that, okay. Yes, thank you. It is going to activate your thirst mechanism, telling exogenously that, you know what, you haven't drank water from morning. Please drink some water. Okay, please drink some water. Then after that, if you exogenously drink water, then your thirst, okay, some water intake has happened. But imagine that is still not enough. Then they will do conditional reabsorption. They'll do what? Conditional reabsorption. Conditional reabsorption of water. Is this clear? Okay. Chalo. Now, ADH loss understood. Same thing goes with RAS mechanism, students. Now, RAS mechanism, what exactly happens? I'm going to orally tell you. So, look here. RAS mechanism happens because there is a JGA apparatus. So, what is going to happen? Your GF has some issue. Okay. Your glomerular filtrate. Okay. Don't think anything else. Your glomerular filtrate has a problem. So when this glomerular filtrate is flowing through the DCT, some cells will get modified to form macula densa. What will it modify to form? Macula densa. This macula densa, okay, this macula densa will send signal to the afferent arterioles, okay, to the afferent arterioles, telling that, you know what, the GF has an issue. So afferent arterioles will be like, don't worry, I will fix everything. I will modify some of my smooth muscles to form JG cells. That is juxtaglomerular cells. These JG cells will release renin. Now we will be like, okay, oh, renin is going to do all the function, chalo, nothing to worry. But renin will be like, no, guys, I cannot do everything alone. So what I'm going to do is, I will go to the plasma, convert angiotensinogen, which is in the blood or in the plasma, which is secreted by the liver, okay, to angiotensin 1. Then we will be like, oh, angiotensin 1 will fix every issue, then no need to take tension. Then angiotensin 1 will be like, no, I cannot fix the issue. I can get converted to angiotensin 2. Angiotensin 2 will fix the issue. Right. Now, when you ask that you want to go out with your friends, okay, what will your mom tell? Go ask your dad. What will your dad tell? Go ask your mom. What will your mom tell again? Go ask your dad. Then what will your dad tell? If mom agrees, you go. Then what will you go and tell your mom? Dad told that if you agree, I'll go. Then you'll be like, if dad agrees, I will agree. So at the end, what happens? You will not go for that party that you wanted to go. You will not go out front. You will not go out. Okay, same thing only. Renin will tell angiotensin. Angiotensin will tell angiotensin 1. Angiotensin 1 will tell angiotensin 2. Clear or not? Okay. Yes. So finally, you will not go out. That is the moral of the story. But here, changes are going to happen. Okay. That is, here, angiotensin 1 to angiotensin 2. Okay. Yes, I have gone through it all my childhood, students. I understand. Okay. I have gone through the same childhood that you are having. Okay. So, but see, this much technology was in there. That's all. Otherwise, everything is very similar. Okay. So, students, angiotensin 1 to angiotensin 2 will not happen chumma. It will use an enzyme called ACE enzyme, which is called as angiotensin converting enzyme. Who is going to give it? Who is going to give it? Lungs will tell, don't worry, I will give you angiotensin converting enzyme. Okay. Now, with the help of angiotensin converting enzyme, we will make angiotensin 1 to angiotensin 2. Now, angiotensin 2 is very powerful. Okay, being a powerful vasoconstrictor, it will increase the glomerular blood pressure and thereby the GFR. Okay, then angiotensin 2 will also activate your adrenal cortex to release aldosterone, and this aldosterone will activate your DCT, telling that please do reabsorption of little sodium and water. Like that, BP also will be fixed. Okay, BP also will be fixed. Now, this also leads to an increase in the blood pressure and the GFR. This is called as angiotensin complex mechanism. Okay, angiotensin, renin, angiotensin, aldosterone mechanism. For those students who did not watch yesterday's class, please go watch. It is clearly explained there. Renin, angiotensin, aldosterone system. This is called as RAS mechanism. Clear? Because this is not the main channel. Mohit has too many doubts in life, no? Actually, one person shouldn't have this many doubts. Technically, in past 30 minutes, he has asked 200 doubts. Yes, clear? You shouldn't ask too many doubts, students. Your body will reject you. Okay, now micturition, that is basically expulsion of urine. Okay, urine formed by nephrons is ultimately carried to the urinary bladder. All of you know this, where it is stored till a voluntary signal is given by the central nervous system. Now, the signal is initiated by stretching of the urinary bladder, and the CNS is going to send the signal. Now, before I go into the next paragraph, all of you, I hope, remembers last class I had taught you trigone of bladder. How many of you remember? This is your urinary bladder. Ratis and Tanuja should remember this. This is urinary bladder. It looks like there's that in Turkey and all they'll do this balloon, no, students. Parachute. Okay, this is your urinary bladder. Yes or no? Now, this, okay, urinary bladder will have 3-4 openings. Okay, 3 openings. 2 openings of the ureter and 1 opening of the urethra. This is called as the trigone of bladder. This is called as trigone of bladder. Now, students, on this urinary bladder, there is going to be a receptor which is called as the stretch receptor. Okay, this I'm just indicatingly drawing it. Stretch receptor. Now, what the stretch receptor is going to do? The stretch receptor is going to send signal to the CNS telling that the urinary bladder has got full. Okay, urinary bladder has got full. So the CNS will in turn send a signal back telling that, okay, do micturition. Okay, micturiration. What is micturiation? Expulsion of urine. For micturiation to happen, there are two sphincter muscles. Okay, sphincter muscle 1 and sphincter muscle 2. Where is it going to be present? Okay, here sphincter muscle 1. Okay, not like that, Mohit. See, again doubt you are asking. Don't ask doubt, I told you just now. No, yesterday's class I did not see you, so I am telling you. Students, this is sphincter. So this is 1, this is 2. So we can call this as internal sphincter muscle, this as external sphincter muscle. Okay. Now students, internal sphincter muscle is not in your control. The moment CNS sends signals telling that let's do micturiation, this will automatically open. This will open. By then you will already get a signal that you have to use the washroom. Okay, that you have to use the washroom. Now since you're already feeling that you want to use the washroom, let's assume your urinary bladder can take up to 800 ml of urine. Okay, 800. So 800 ml of urine it can take. That means around 500 ml only you will have the tendency. Then you will hold up to 800 ml. Now by the time it reaches 800 ml, now you go to the washroom and you decide when to do micturiation. That time only you will be able to open this. That means it is in your control. It is going to listen to you. Okay, this is going to be voluntary. Clear? Yes or no? So this is how it happens, students. Now how the urine is going to be passed? It is going to come from the collecting duct. Okay, collecting duct to renal pelvis. All of you remember, if you are able to decode this. Then to ureter, ureter to urinary bladder, urinary bladder to urethra. Students, don't think that ma'am's head got bent, but yeah, this is comfortable for me, and if I move that side, it's paining. Okay. Yes. So students, collecting duct to renal pelvis. This is the passage of urine. Okay. This is the passage of urine. Got it? Passage of urine. Got it? Clear or not? This is. So from collecting duct to renal pelvis to ureter, ureter to urinary bladder to urethra, and then your micturiation happens. Okay. Micturiation happens. Now there are some numbers that you need to remember. Okay, now look here. Here, to initiate the contraction of the smooth muscles of the bladder, a simultaneous relaxation of the urethral sphincter should happen. Okay, now listen students, very simple. If urinary bladder has to open, okay, that means not open, like expulsion should happen. This is your sphincter muscles. What they should do? They should relax. What should a urinary bladder do? They should contract. Okay, they should contract. Understood? Yes or no? Is it? Oh, just tell me how she's getting rid of this. Okay, because it's very painful. It's very painful, and I'm not able to. No, it's not able to be because of the pain. Yes. So students, usually it happens for me, but it goes very soon. But today it has stayed like from yesterday night till today morning, today evening it's there. So we have to see what happened. So students, here what is going to happen? Your sphincters, okay, sphincter muscles have to relax. They have to relax. But your urinary bladder has to contract. Okay, urinary bladder has to contract. This moment you remember, okay, this will become easy. So urinary bladder contracts, okay, sphincter muscle, this is the sphincter muscle, it has to relax. Urinary bladder has to contract. So let's say this is the system, urinary bladder contracts, sphincter muscle relax, micturiation is going to happen. Okay, micturiation is going to happen. Now the process of release of urine is called as micturiation, and the neural mechanism causing it is called as micturiation reflex. An adult human excretes on an average of 1 to 1.5 liters per day. The urine formed is light yellow colored watery fluid which is slightly acidic. That means 6. Okay, 6. It is not neutral, but it is slightly acidic. Okay. On an average, 25 to 30 grams of urea is excreted out per day. Students, this is the important PYQ. Make a note who are going to write NEET exam. This is a very important PYQ. On an average, 25 to 30 grams of urea will be excreted per day. Per day. They can ask you for per minute. Okay, you have to do maths. Now, various conditions can affect the characteristics of urine. Analysis of urine. So, students, before you remember, when we were in small age and all, we used to do a lot of urine tests. Why? To just check if you have, any urine infection, if you have anything, kidney is functioning normally. We used to do urine, test. Nowadays, it has come a little less, but then still, it used to be one of the preliminary tests, or let's say a primary diagnosis used to happen because of the urine test. Why? Because the doctor will be able to understand if the kidney is working normally, if there is correct amount of urea, if there is correct amount of ketones, anything is too much because of our eating habits, some addition of fats is there. All this they'll be used to preliminarily understand because of the urine test. But nowadays, because of the kind of diseases that we have, we also do blood test a lot, right? So, for example, presence of glucose will be called as glycosuria, and then ketone bodies is going to be ketonuria. Ketonuria in the urine, which is going to cause diabetes mellitus. Okay. Now, role of other organs in excretion. So, it's not just a kidney. Okay, it's not just a kidney who is going to help in excretion, but there are other organs like kidney, lungs, liver, skin, who is going to help us with excretion. Our lungs remove large amount of carbon dioxide. Did we study this in breathing and exchange? Yes, ma'am, we studied it in breathing and exchange, that there is large amount of carbon dioxide that is going to be excreted out, and also significant quantities of water every day. Liver, the largest gland in our body, secretes bile containing substances like bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, drugs. Most of these substances ultimately pass out through the digestive waste. Now, when we eat a lot of tablets, students, where is it going to go? Where is it going to go? It is all going to go into your liver. So whatever is left over will be excreted out through the help of digestive fecal matter. The sweat and the sebaceous gland. The sweat and the sebaceous gland in the skin can eliminate certain substances, through their secretions. Sweat produced by the sweat glands is a watery fluid which contains salt. Small amounts of urea will be there, lactic acid will be there. Like if we are doing a lot of exercise, students, then lactic acid will come out. Though it is the primary function of sweat to facilitate a cooling effect on the body surface, it also helps in removal of some waste mentioned. Is this clear? Chalo. Sebaceous glands eliminate certain substances like steroids, hydrocarbons, waxes through the sebum. This secretion provides a protective oily covering for the skin. So now imagine, see, now after taking a lot of class, like I had DNA also, now when I do this, I will get a lot of oil on my hand. Why? Because this is a kind of protection. So when whatever like dust particles I was exposed to, the oil glands are going to throw it out. The oil glands are going to move the little wax, steroids or dust particles outside. Understood? That also leads for pimples as well. Sometimes what happens when we don't take care of our skin well, like there's so much oil secretions here and you're not washing your face after that, what exactly happens? This portion will start holding the dust and accumulating it, and you will get acne and pimples. So that's how you get it. So you should always wipe your face at least with a wet cloth or something when you're coming from outside, because there'll be a lot of dust which has gone and fallen on off your skin, and it will go and settle inside the pores. Wow. Now, malfunction of kidney can lead to urea in the blood called as uremia. So students, very simple conditions. Glucose is more. Glucose is more. I think if I write it here, you'll not be able to see. Glucose is more. Glucosuria. Ketone bodies are more. Ketonuria. Urea is more. Uremia. And students, when such condition comes, you have options like hemodialysis. Hemodialysis, where you will remove the blood, you will take the blood, the dialyzer will take the blood, and that blood will be conditioned and purified. Purified. Now students, for that to happen, you should remember that there is a cellophane tape. There's a cellophane tape. Now this cellophane tapes are going to have small pores, and they will keep it inside a dialyzer. Okay, and inside this you're going to see the dialysing. Thank you, Dikshi. Inside this you're going to see the dialysing fluid. Okay guys, all of you are going to be there. No, Sunday I have my Navratri series. Don't go off somewhere. I'm cancelling one plan and doing the series for you people. So if you people are not there, I'll be very disappointed. Okay, dialysing fluid. Got it? Dialysing fluid. Now this dialysing fluid will have 0%. Okay, absolutely no nitrogenous waste. Okay, absolutely no nitrogenous waste. Understood? No nitrogenous waste. Why nitrogenous waste shouldn't be more? Because students, the blood that we are taking from the person, that person's blood is rich in nitrogenous waste. No, that's why we're doing purification, because kidney has failed. Renal failure will cause hemodialysis. Okay? Yes. Z also is fine, S also is fine, no problem. Okay, that is British and all this American English, that does not affect Indians. Okay, so students, here what is going to happen, since here in this dialysing fluid there is no nitrogenous waste, nitrogenous waste is going to move from a region of higher concentration to a region of lower concentration. Okay, with the help of normal diffusion. Okay, with the help of normal diffusion, that means your urea which is in the blood, okay, urea in the blood is going to come out. Okay, is going to be going into the dialysing fluid. So later the blood will be taken in. Now when we are taking the blood, we will add heparin, which is an anticoagulant. Okay, because body naturally makes it. When we take the blood out, it will immediately start coagulating. Okay, they will start coagulating. So to avoid that, we will add heparin. Okay, then when the blood is going back, okay, when we are adding the blood back to the person's body, we will add anti-heparin. Okay, anti-heparin. Clear or not? Yes. This is how dialysing fluid works. This is your cellophane tape. This cellophane tape, students, will have small pores which will allow the passage, okay, up and down movement of it. The cellophane tape. Yes. Okay. In school, no, when I was in 10th, I have done this experiment. Matlab, I have done this for exhibition and we got first prize. Okay, so how we did it with bottles, okay, with those straws and tubes and then syringe, with using all of this we had done it. Okay, so urea can be removed by the process of hemodialysis. Blood drained out from a significant artery is pumped into the dialysing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tape surrounded by a fluid called as dialysing fluid, having the same composition as that of the plasma except the nitrogenous waste. So the porous cellophane tape of the tube, okay, no exhibition, no lab works. If this happened in 10 students, in 10th it happened. Okay, we'll have an inter-school competition and then we'll have intra-school competition and people from outside will come and judge us. Okay, allows the passage of molecules based on the concentration gradient. Okay, as the nitrogenous base are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. Understood? Excuse me. Okay, the cleared blood is pumped back into the body through a vein. Okay, so with the renal artery we are taking the blood. Students, here is the catch. Here is the catch. Renal artery will have what blood? Let me see who is going to answer this. Yesterday I had mentioned this. Renal artery has what blood, students? Oxygenated, deoxygenated. Happens. Tell me, all of you, in renal artery, what is going to be there? Let me see my smart kids. Renal artery will have what kind of blood is the question. Hello, answer fast. See, renal artery is going to have oxygenated blood. Yes or no? Oxygenated blood, but toxic blood with toxic substance. Yes or no? Agreed, all of you? This is what I told you exactly yesterday. So we have taken oxygenated blood, but that blood is rich in waste materials, urea. Now which blood are we going to give back? With the help of renal vein. Okay, after purification we are going to give them deoxygenated blood. Okay, deoxygenated blood, but with no toxins at all. Okay, no toxic nitrogenous waste. So here always remember, oxygenated, deoxygenated totally means, okay, totally means that you are talking about the oxygen and the carbon dioxide content. You're not talking about anything else. Okay, please understand this. Whenever you're talking about oxygen and your carbon dioxide, that is only when you will talk about oxygenated and deoxygenated. Otherwise, you're definitely changing the conversations here, because here it is oxygenated plus rich with toxic substance. Got it? Yes. Okay, next one. After adding antiheparin to this method is boon for thousands of uremic patients. So students, this is usually done when they have a renal failure. Okay, even if one kidney is there, okay, even if one kidney you have sold to buy iPhone, one more kidney, it will work. Okay, but that does not mean you will sell kidney for buying iPhone. Anytime that kidney you do not maintain properly, you have drinking habits, any of the issues are there, or you have some genetic disorder, then this kidney, second kidney, can fail easily. So that's why God has given two kidneys. Okay, don't question God's decision. Now kidneys transplantation is the ultimate method. Students, for a long run you cannot do dialysis. It's a very expensive thing, and also nowadays because of all the health insurance and other things, yeah, maybe it will be a little less. Okay, little less, but it is not something that you can take for a longer period of time. And also there are situations where, you know, because the person's, you know, the electrolytes in the body have changed, the concentration of the electrolytes has changed, the person will start having nose bleeding, and the person will be admitted, and they will tell that you need to do a transplant. Okay, you need to do a transplant. Yes, both the kidneys are required. Okay, so students, renal failure, okay, renal failures, this, if it happens, you have to take kidney from a close relatives of yours, so that your body does not reject it. Okay, so nearly, preferably a close relative, to minimize the chances of rejection by the immune system. So modern clinic procedures have increased, like we use a cyclosporin A. Okay, we'll use cyclosporin A, which is an immunosuppressant, which is going to help with graft rejection. Okay, reduce graft rejection. Okay, got it. Now renal calculi, students, very common disease, kidney stone. At least once you would have heard in your life, right? Kidney stone. That is because a stone or an insoluble mass of crystallized salt, like your oxalates, are formed in the kidney. Again, this is also because of the changes in the electrolyte in your body. Okay, electrolyte in your body, and because of which you have also, it could be a main reason that you have not consumed enough water for expelling out of this, you know, electrolytes. So what they will do, they will accumulate and form stone-like structures. Okay, sometimes, usually majority times, let's say 95% time, the doctor will give you a medicine and they will expect you to drink lot of water. Okay, usually renal calculi or your stones will go. Okay, stones will go. But during a situation where your stone is bigger, you will have to go for a surgery. Okay, now glomerulonephritis, students, it is the inflammation of the glomeruli. The glomeruli, you know, which we spoke about, that inflammation happens in the blood vessels, in the capillaries, that will lead to glomerulus. Okay, glomeruli. This can change your GFR rate, because if there is an inflammation, then you know that the flow of blood is going to be difficult. Okay, so inflammation can cause a trouble. Okay, inflammation can cause a trouble. Is this clear to all of you? Yes or no? Yes? No doubts, right? If you have any of the doubts, please ask me right now. Okay. So the most important things to focus in this chapter is, first of all, you have to know the difference, which is kind of simple. You have to know the human excretory system You have to know the order, how the blood is flowing, how the urine is flowing. Okay, human excretory product. Then students, urine formation 3 steps. Okay, urine formation 3 steps. For NEET students especially, I'll tell you your RAS mechanism. Lot of questions have come. All the mechanisms. Okay, if you're a board student, then all the headings are important for you. If you're a NEET student, your mechanisms are very, very important. Okay, what is secreting, from where is it secreting, what is the function, all these questions will keep coming. Okay, and dialysis also 1 or 2 questions, but not again, not a major chunk is not going to come. Dialysis 1 or 2 questions are going to come. Okay, we take the medicine until the signs are down. Okay, sometimes what happens, the body will start rejecting, and there is going to be sometimes bleeding, sometimes it is not going to accept. So until the symptoms are down, the doctor will be giving 1 dose. Okay, obviously 1 dose they'll give, and they will check. Okay, then they will check the duration, because different bodies, all of our bodies are very different, and we cannot predict at what dose we can give for each of them. Okay, so the doctor is going to check, and they will understand. I have heard it can go up to even 1 week. Okay, 1 week, until the body accepts it, it can even go up to 1 week. Okay. Mohit, in JE there's no biology. Okay, I don't know what to tell you. He's asking in JE how many questions will come from this chapter. What did you think? This was maths, sir. This is biology, dear. There is no questions, and you're thinking about JE means. Done? Okay. Yes, we are done for the day. Okay, so thank you so much for joining, students. See you all in next class, and I hope all of you enjoyed the session. Yes, stay healthy, stay happy, and thank you so much. Tata, bye bye.